With new technology being discovered and researched every day, it will be a matter of time before it will become possible to treat diseases that once had no cures. One of the most researched forms of biotechnology are stem cells, with a seemingly infinite amount of potential applications. To understand stem cells, it's extremely important to have a knowledge about what cells are and how cell reproduction occurs. Often referred to as the building blocks of life, cells are the most basic units of matter to be considered a living organism. The first cells were discovered in 1655 by Robert Hooke as he examined the cork of a wine bottle. After inspecting the cork with a microscope, he found minuscule structures and referred to them as cells. However, he was not able to see individual organelles such as a nucleus or mitochondria that can be seen today. The first living organisms viewed through a microscope were bacteria in water, discovered by van Leeuwenhoek in 1678. Research of cells was continuously conducted, and in 1838 the first cell theory was created. The claims present were tweaked and modified to result in the modern cell theory. All living organisms are created using cells. Cells are the basic functional unit of life. New cells are created from previously existing cells through mitosis and meiosis. Cells contain the genetic information that defines us in the form of DNA and chromosomes, and cells are nearly identical in chemical composition and organelles, and that all energy transfers and flows in life occurs within or amongst cells. Cells can be classified into two main types: prokaryotic and eukaryotic cells. Prokaryotic cells were the first life on Earth. These cells are simpler forms of life, being mostly unicellular organisms between 1 and 10 micrometers. The DNA of prokaryotic cells are in the form of circular, naked forms with no added proteins or substances. It's located in the non-membrane bound region of the cell referred to as the nucleoid. According to the theory of endosymbiosis, eukaryotic cells first appeared as mitochondria and chloroplasts took permanent residence in larger prokaryotic cells. This theory proves why mitochondria and chloroplasts have double membranes rather than one, like other organelles such as the endoplasmic reticulum and the Golgi bodies. Moreover, it also explains the fact that mitochondria and chloroplasts have circular DNA and reproduces independently from the rest of the cell. This diagram shows how the mitochondria and chloroplasts entered and took permanent residence in the cell. Eukaryotic cells are much larger than prokaryotic cells, ranging between 10 and 100 micrometers in size. The two most common forms of eukaryotic cells are animal and plants. The largest difference between animal and plant cells are the organelles present. Plant cells have a cell wall that provides structure and support to the cell. Furthermore, plant cells have a chloroplast that use sunlight energy and carbon dioxide to produce water and oxygen through the process of photosynthesis. Plant cells also have a large vacuole in the cell that can occupy up to 90% of the cell's volume. The vacuole acts as a storage facility that can store nutrients as well as store unwanted substances. The size of individual cells depends largely on the cell's specialization or role. Our body has diverse cell types, ranging from nerve cells to muscle cells to fat cells that all have different sizes. As previously mentioned, the cell theory states that all cells are derived from pre-existing cells. A cell's life is known as the cell cycle and consists of three main functions: growth, repair, and reproduction. There are two methods used by cells for division and the creation of new daughter cells: mitosis and meiosis. Mitosis is used in the vast majority of our body cells and produces two genetically identical daughter cells that preserve our body's diploid number, 23 pairs of chromosomes. On the other hand, meiosis only occurs in the sexually reproducing cells of our body, the sperm and egg. These types of cells are also referred to as gamete cells. Meiosis produces four haploid cells with 23 chromosomes that are genetically diverse. The sperm is the haploid gamete cell of a male, and an egg is the haploid gamete cell of a female. Each month, an egg is produced and grows in a female's ovaries. Once matured, the egg enters the fallopian tube. This whole process is called ovulation. When a sperm reaches an egg in this period, the surface and genetic makeup of the egg changes and fuses with the sperm to prevent any other sperm from entering the egg. When the sperm and egg meets, a diploid zygote is produced. After approximately 24 hours, the first cell division occurs and the embryo is referred to as a morula. As the morula travels in the oviduct, cells undergo rapid cleavage divisions. The cells produced, known as blastomeres, are all undifferentiated at this stage. These types of cells are referred to as stem cells, or cells that have varying potency that can self-renew and become specialized into various different forms. Approximately 5 days after the development of an embryo, an outer layer of cells is separated from the inner cell mass. Cells in the ICM can become any cell type, but after implantation to the uterus, the large majority of these embryonic stem cells are depleted. Contrary to the embryonic stem cells that are pluripotent and have an unlimited capacity for cell renewal, adult stem cells are multipotent. Adult stem cells have lower potency than embryonic stem cells, or a lower number of possible fates of the cell. Adult stem cells are derived from adult tissue and are believed to have a limited ability of differentiating into diverse cell types. As adult stem cells are found in adult tissue and do not involve the destruction of an embryo, the technology is not considered to be ethically controversial. Although embryonic stem cells have a higher potential due to the fact that they can differentiate into more types of cells, and due to the fact that they can be easily grown in culture or in labs, there is more ethical controversy due to the utilization of an embryo. Religion is one of the main issues that affect one's views on stem cells and its ethics. In a survey conducted by Pew Research Center, 52% of those opposed to stem cells had religion as their biggest factor contributing to the decision. For example, believers of Judaism support stem cell research as they believe life starts after 40 days of conception. For these people, 5-day-old embryos are not considered to be living beings. Meanwhile, Roman Catholics oppose stem cell research due to their belief that life commences at conception. Stem cell research is generally supported in Muslim communities as well due to their belief that life begins approximately 40 days after conception when the soul enters the human body. The following table also represents other religions and its views towards stem cell research and application. Different countries have widely different regulations on stem cells as well. In Germany, the 1991 Embryo Protection Act restricts the destruction of embryos to research. Although research on stem cells was illegal in France, the French National Assembly passed a new law in 2013 allowing research on embryonic stem cells to be done in specific circumstances in which the medical purpose of the research and necessity of an embryo is clearly stated. Meanwhile, being one of the leading nations of stem cell research in the world, South Korea promotes stem cell research and the therapeutic cloning of embryonic stem cells. The list of potential applications of stem cells is endless. As of January 2016, only 10 stem cell applications have been approved worldwide after testing on animals and on humans. However, this number is expected to drastically increase due to the fact that lots of research is currently being done about this topic. Some possible aspects in which stem cells can be used are diabetes, severe burns, bone marrow transplants, Alzheimer's disease, and the Parkinson's disease. The following diagram shows a possible application of stem cells to treat burns. By taking the adult stem cells in the body and growing it in culture or in a laboratory, it will become possible to apply the large amount of stem cells created back to the region of the burn.
당뇨병이란 우리 몸에 인슐린이 부족해서 생기는 병입니다. 당은 우리 몸에서 영양소, 즉 휘발유와 같은 역할을 합니다. 중요한 그 휘발유와 같은 그런 역할을 하는데 그 당은 우리 몸에서 휘발유와 같은 역할을 하는 데 있어 가지고 인슐린이 있어야만 그 혈액 속에 있는 당을 세포 속으로 운반을 하는 것입니다. 다른 말로 하면은 인슐린이 부족하면은 혈액 속에 그 당이 세포 속으로 활용되지 못하기 때문에 들어가지 못하기 때문에 혈액 속에 당의 양이 많은 것을 당뇨병이라고 합니다. 그래서 인슐린이 아예 만들지 못하거나 인슐린이 적거나 인슐린을 활용할 수 있는 인자가 적을 때 그 혈액 속에 있는 여러 가지 당이 활용하지 못하는 게 뇌로 나오게 되는 것입니다. 그걸 당뇨병이라고 하는 것입니다.
Now, there are no cures for diabetes. Instead, the ultimate goal is to control blood glucose levels by changing one's diet, losing weight, and exercising. Some treatments are also given, such as metformin and sulfonylureas. Metformin causes body tissues to become more sensitive to insulin, increasing the effectivity. Sulfonylureas aids the excretion of larger amounts of insulin. However, these two medications have possible side effects depending on the patient's underlying health status and other health issues. The cost of medication can also be a burden for some patients.
최근에 그 줄기세포를 이용하여 당을 치료할 수 있는 연구가 많이 활발되고 있습니다. 줄기세포의 췌장 세포를 우리 몸에 이식함으로 인해 가지고 그 췌장 세포의 베타 세포가 활발함으로써 당을 근본적으로 치료할 수 있는 길이 연구되고 있으며
Patients' biliary tree. The usage of these types of stem cells have a higher success rate due to the fact that they are precursor cells, which mean that they are partially differentiated into the beta cells of the pancreas. Stem cells can be used to regenerate tissues and boost the body's ability to produce insulin. The beta cells of the pancreas are responsible for the excretion of insulin. The red regions of this animation portray the beta cells and its locations within the pancreas.
최근에 그 한국에서는 그 줄기세포를 이용한 그 베타 세포를 쥐에 콩팥에 이식하여서 그 쥐가 인슐린을 분비함으로 인해 가지고 그 아무 부작용 없이 베타 세포에서 인슐린 분비되는 데 대해서 성공하였습니다. 그래서 쥐에 성공한 이 연구를 1년 후에는 사람에게 직접 인용하여서 당뇨병 치료에 큰 발전의 전기가 되지 않겠나 생각됩니다.
After the discovery of cells in the 17th century, continuous research has been done to produce numerous new discoveries. One of the most innovating discoveries may be stem cells. Stem cells have infinitely many applications in the treatment of diseases that were previously incurable. The ability of stem cells to specialize into different types of cells increases its potentials for use. One of the developing applications of stem cells are the treatment of diabetes. By injecting one's own stem cells into the pancreas, it may become possible to replenish the pancreas with functioning beta cells that can provide a cure to diabetes by increasing the production of the hormone insulin.